Good afternoon. Welcome to the University of Western Australia's Institute of Agriculture 2022 Postgraduate Showcase. We call Frontiers in Agriculture because that's the cutting edge research done by the PhD students who are going to talk today. Now, that's the one. So before we getting started, it is customary for the university to acknowledge that the campus is situated on Noongar land, and the Noongar people remain the spiritual and cultural custodians of their land and continue to practice their values, languages, belief, and knowledge. I would also like to acknowledge there are many people here, but certainly Terry Enright, the chair of our Industry Advisory Board, John Higgs, a number of colleagues from DPERD here, and some of them have been regular. For example, Darshan Sharma has been 16 years coming, so we'll be giving an extra glass of wine this afternoon. But also, more importantly, I would like to welcome the new team joined at the Murdoch University under the leadership of Professor Rajiv Washni. Rajiv, you can raise your hands. And also Anu and Vanika and of course Abhishek is not here. So you're most welcome. The first thing you need to remember is that universities and people do collaborate. Just ignore what people say. Whoever you want to collaborate, go for it, and vice versa from this university. And of course, John Hamblin and Paul Fox. I haven't met Paul Fox for a long time. Paul Fox was a PhD student here under Roger Boyd, and then he worked in CIMET, Mexico for a long time, and then he came back and became crop science manager of the Australian Centre for International Agriculture Research for nearly 5 years. Then he was in Bangladesh. So he lives in Adelaide. Welcome, Paul. These are all not in my notes, so you must be wondering where it's coming from. And more importantly, distinguished guests, university colleagues, media, students, and ladies and gentlemen, we expect more people will come. People will come and go depending upon their interest. And a very warm welcome to our annual Postgraduate Showcase, which is the 16th event. It has been a very long time we have been doing that. About 6 to 8 PhD students are selected based on the recommendation from the heads of the schools and the supervisors. And this is since 2007 when we re-established the UWA Institute of Agriculture. A little bit about the ranking. I know that in today's paper there's a lot of message that ranking is baseless, ranking is not important. Ranking is important. It is just a benchmark. It is not the end of everything. But in the academic ranking of world universities in 2021, UWA is ranked 16th in the world and #1 in Australia in agriculture. So the UWA Institute has played a significant role in that. More importantly, in this context, the postgraduate students, PhD students, must publish their work, and we make sure that many of them publish before they're finishing their program, and those papers also contribute to the ranking. So what's the Institute doing? Those who are not very familiar, basically integrating research, education, training, and community engagement through communication in agriculture and related resource management areas. So the Postgraduate Showcase brings together UWA some of the best PhD students who has at least completed 50% of their PhD, mostly 75% are ready to submit within the next 3 to 6 months. So this is really a matured project, and so you will see some interesting aspect. The idea of this Postgraduate Showcase is not really showing a lot about science, but synthesizing that science in such a way that there are farmers and general audience can understand. It gives a training to the PhD students as part of their training program. As I mentioned, this afternoon we will hear from 7 PhD students or 6? 6. So the presentations are divided into 2 sessions. One of the students has to pull out due to some medical emergency. The That's all I was supposed to do. So the first session is chaired by Dr. Hayley Norman, and Hayley is here. I'll just say a few words about you. It's my pleasure to introduce the chair of the session, Hayley Norman. Hayley is the senior principal scientist at CSIRO here in Floreat, and a member of the UWA Institute of Agriculture Industry Advisory Board. Hayley is passionate about helping industry achieve profitable and environmentally sustainable farming systems in low rainfall Mediterranean type environments. After gaining her PhD, which I remember, in the reproductive ecology of annual legumes from UWA under the supervision of Professor Phil Cox, Hayley moved to CSIRO to conduct farming system research with a focus on livestock production with mixed farming systems. She developed an interest in use of drought and salt-tolerant native shrubs, including saltbush, and led the team that developed Anamika saltbush, one of the varieties selected from where do you think? Hayley enjoys collaborating with farmers and industry to develop farming system. She has worked on agricultural practices and projects in Iraq, Syria with ICADA, Afghanistan, and Tibet. She sits on the Western Australian Livestock Research Council, the WA Crawford Fund Committee, and chair of the WA Soil and Land Conservation Council. Please welcome Hayley.
Thank you, Professor Siddique. I have the pleasure of introducing our first postgraduate presenter, MD Khairul Islam, from the School of Allied Health. Khairul completed a Bachelor of Pharmacy and Master of Pharmacy from the University of, Rajasthani, Bangladesh. Is that good? Rajesh. I'm much better with my Middle Eastern languages. After that, he worked as a product executive for 2 large pharma companies for about a year before changing his career towards academia. He worked as a lecturer at the Department of Pharmacy at Perennial University in Bangladesh for 3.5 years before moving to Perth to complete his PhD at the University of Western Australia. Previously, he was involved in research on food microbiology, and now he is working in the field of medical chemistry in the area of authentication and quality control of honey at the Division of Pharmacy. His research is funded by the Cooperative Research Centre for Honeybees and honeybee products. Khairul will be presenting today on exploring high-performance thin-layer chromatography as a novel and analytical approach for honey analysis. Please welcome Khairul.
Hello and welcome everyone. Thank you for the nice introduction. So today I'm going to present a part of my PhD project. First of all, I would like to thank my principal supervisor, Associate Professor Dr. Connie Locher, and my co-supervisor, Professor Leong Lim and Catherine Hammer. I'm a member of Cooperative Research Center for Honeybee Product, and I wish to thank for their funding support for my PhD project. Before going deep into the honeybee industry of Australia, first of all, we all know honey is sweet, viscous, highly complex natural substances produced by the bees from the flower nectar. Bees collect the nectar, transform into honey, store in their hive, from where the beekeepers collect the honey for us. Honey is mainly consists of sugar, especially the major sugar like the glucose and fructose. Apart from that, a little bit of sucrose and maltose and other oligosaccharides also present. Honey contains a large number of simple phenolic and flavonoids. Apart from that, a little bit of or small quantities of protein, amino acid, minerals and vitamins also present in honey. Australian honeybee industry worth about $100 million through the direct selling of their honeybee product like honey, pollen, propolis and waxes. Additionally, it contributes about $14.2 billion to the economy through the pollination service and also the tourism or the recreation industry. Today, I am going to present or highlight how my research is helping the honeybee industry to develop high-value product for the customer, especially in Australian context and broader international context. So honey differs from each other in terms of their organoleptic property like the taste, smell and flavor, as well as some bioactivity. Those organoleptic properties and the bioactivities are closely related to their phenolic and flavonoid compounds. Phenolic compounds are mainly responsible for their antioxidant activity, antibacterial, anti-inflammatory activity. On the other hand, flavonoid contributes to their especially organoleptic characteristics as well as some health benefits. As you all know, the price of honey differ really vastly due to their floral sources. So it's a really great interest for the industry to authenticate the nectar source or the floral source of honey. There are lots of methods available out there to analyze the honey for authentication as well as quality control. Especially the first one is pollen analysis, commonly known as melisopollenology. The common approach is that identifying and quantifying the number of pollen, we might conclude that the bees collect their main nectar from that particular plant. The second approach is identifying and tracking of the marker compounds and the metabolomics using costly and complex instrumentation like HPLC, GCMS or NMR. Thirdly, the quantification of the measured sugar, especially the fructose to glucose ratio, which might act as an authentication tools for honeys. But all of these methods has their own drawback and might not suitable for the Australian honeybee industries. So, as a part of my PhD, I have developed a novel and cost-effective analytical tools using one of the novel instrumentation called high-performance thin layer chromatography, in short HPTLC. So, as HPTLC is the core of my research, so I will discuss a little bit about it. HPTLC or high-performance thin layer chromatography work exactly like thin layer chromatography or TLC. So, in principle, if we apply a complex mixture of color in a paper strip and dip the paper strip in a water, due to the capillary action, the water starts moving out. Along with it carries the complex color mixture. Due to the chemical differences of the colors and also the interaction between the color, water and also the paper strip, they start separating from each other. At the end of the analysis, we have find the colors are separating from each other. Obviously, for honey analysis, we are not going to use the color. For honey analysis using HPTLC, we usually use honey solution or honey extract. Obviously, we are not going to use the water as a solvent. We use specifically designed mobile phase for a specific purpose. Instead of paper strip, we use glass plate evenly coated with silica gel, what we refer as HPTLC plate. So, here is an example how HPTLC floral fingerprints look like. In here, we apply the floral source of honey. To determine the floral source of honey, we extract the honey with an organic solvent because the bulk of the honey is sugar and sugar has nothing to do with their floral source. So, we extract the honey with organic solvent and then apply the extract at the bottom as a single band. During the analysis, the solvent system carried the extract and separated them along the way and producing individual bands, which we can analyze in different light conditions. In this case, as 366 after derivatization. One thing to note that not all the compounds are analyzable or visible in single light condition. So, we have to analyze them in different light condition, especially in this case at white light. Here are another two honeys. That means in these two sets, there are three different honeys, beekeeper named as Jara, Banksia and Mary. You can visually see that their fingerprint, their banding pattern are different, and which we argue that is closely related to their floral sources. Obviously, we are not going to conclude based on just single sample analysis. Throughout my PhD, I have analyzed more than 500 different Western Australian honeys to come up with this simple analytical technique. Here is some example. there are 6 different honeys beekeeper named as Jera, and we analyze with the help of HPTLC. After the analysis, you can visually see that they have common banding pattern. We can conclude or we can summarize all the banding pattern and can refer to as HPTLC fingerprinting, which we argue that is reflective to their floral sources. Now, we have also developed how we can help this HPTLC fingerprinting to help the industry to promote their product, because ultimately this is our goal, to help the industry with the backup of science. So here we have analyzed quite a significant number of Western Australian iconic Mary honey, and looking into all of their fingerprinting and summarize that at the light condition 254, they have 2 distinct band, 366 one, white light 2 distinct band, and 366 after derivatization 3 distinct band. So we have summarized this, and the beekeeper can use this as a marketing tool. We have also helped them to develop or vision how they can translate this information to communicate with the customers. So we take one of the fingerprint that's visually really startling or stand out. We converted this fingerprint into artistic representation and inserted into the jar label. Also, we have added a QR code. So when the customer is going to buy the honey, they can scan the QR code, which will lead to a specific web page that will show the exact fingerprint along with the science behind it. This is another approach how we can communicate with the customer through the backup of science. Until recently, no one has analyzed quite a simple or developed quite a simple method using HPTLC for the analysis of major sugars in honey, especially the glucose, fructose, sucrose, and maltose. You can see as a part of my PhD, I have developed a novel and really cost-effective method for the analysis of major sugars. I have applied this method for the quantitative dimension to detect and quantify how much major sugar present in honeys. Here is an example where we use this method to quantify the sugars present, especially the glucose and fructose present in Western Australian iconic Jera honey. Sometimes a lot of people, especially the fraudulent people, add or mixture cheap sugar syrup to bulk up the volume of honey, ultimately the profit. They use mainly the sugars of glucose syrup, golden syrup, corn syrup, or maple syrup. To track the addition or adulteration of those syrup, I have used my sugar analysis method first to analyze those individual sugar syrup and find out that apart from the major sugar, especially the glucose, fructose, there are excess additional quantities of maltose and sucrose also present. So we can easily identify and quantify those sugar to know their adulteration status. As a lab setup for post-harvest adulteration, I have admixture known quantities of these 4 syrup into one of the iconic Australian or New Zealand honey, Manuka honey, and you can see the Manuka honey only contained noticeable quantities of fructose to glucose, but at the 30% concentration of these 4 syrup, you can see the additional bands are present, which easily can confirm their adulteration status. Due to the ease of operation, cost effectiveness, and also the convenience to the industry, CAMAG, the world leader of planar chromatography, has adapted the sugar analysis method I developed into their HPTLC Pro system. And this is an example how my research is helping the industry to translate their research. Now, the honeys are priced based on their different characteristics. So customers usually mainly focus on their antibacterial or antioxidant activities. So antioxidant activity can be quantified with a different number of assays, especially the DPPH assay, that's known as 2,2-diphenyl 1-picrihydroxyl assay, or FRAP assay, that's ferrous reduced oxidant assay, or total phenolic content. For my PhD, I will mainly focus on the DPPH assay. One of the drawback of the conventional DPPH assay, it will give us the total quantity of the antioxidant activity. It can't distinguish between the two honeys that might have similar DPPH activity, but different compounds are responsible for those activities. So as a part of my PhD, I developed a novel analytical approach for the antioxidant activity using HPTLC. So to do that, we again extract the honey, and then we analyze the honey using HPTLC. During the analysis, the individual compounds are separated from each other, and then the HPTLC plate we treated with the DPPase. DPPase is a powerful oxidative compound which vigorously react with any antioxidant substance. When they react with antioxidant substance, they changes their color, and we can trace the color and quantify the antioxidant activity. In this picture, the DPPase has an inherent color or pink. You can't see any band because there is no antioxidant compound present in this plate. But we use gallic acid in different quantities. Gallic acid is a known standard commonly used to express the antioxidant activity, especially DPPase antioxidant activity, as gallic acid equivalent. So we have applied in different quantities, and you can see it react with the DPPase and produces single band. We have used the we have used the gallic acid on top of the manuka extract. You can see apart from the DPPase, there are additional bands are present in here, which confirms that the manuka honey contain those compounds that are antioxidantly active. This method is really helpful for distinguishing between the honeys that have the similar total activity, but different compounds are responsible for this activity. Especially in this case, iconic Mary honey and multifloral honey. Mary honey have 2 different, and the multifloral have 3 different groups of compounds are responsible. 4 different groups of compounds are responsible for those activities. So summary, as I can summarize everything, that how HPTLC is going to help the industry to promote the honeybee product. HPTLC has a lot of advantages, and also it can cover a wide range of application, especially for the identification of the floral sources, the detection and quantification of the major sugar present, which ultimately can trace their adulteration status, and finally to track the antioxidant activity of the honey. Apart from all of these, the HPTLC come with some of their inherent advantages, like it's easy to perform, and the data interpretation is really easy. It's really cost effective because it takes a little bit of solvent, a small amount of the sample to perform the all experiment. So it's environment friendly and also I think sustainable too. Finally, one instrumentation can offer a wide range of analytical technique for the analysis of honey, which can be done in a single lab setup. So it ultimately be a suitable routine analytical industry technique for the honeybee industry. Thanks everyone.
Does anyone have any questions for Khairul? Phil?
Interesting things. I'm actually interested in what if you found out to be, not that I do this, but if you found out sort of whacking in a bit of golden syrup into stuff, what is the penalty at an industry level? What if you're caught doing that sort of thing? What happens?
Actually, it's as a researcher, we're developing the tools.
Yeah.
It's up to the industry and the regulatory bodies how they are going to use the tools and counteract that type of fraudulent behavior. So that's not actually up to us to decide. It's up to the regulatory body how they are going to react with that.
Yes.
So in addition So in addition to know what components in honey, are you able to quantify the amount of those components?
Okay, so especially for the floral fingerprint, so individual compounds are separated from each other. My PhD is mainly focusing on the pattern of this banding. I am not mainly focused on what the compounds are. There is another PhD project going on under the umbrella of Cooperative Research Center for Honeybee Product. My colleague Ivan, he is looking into specifically what those compounds are. And he has already identified quite a few. And you can visit to the Cooperative Research Center for Honeybee Product website. You can find a lot of information over there.
Thank you.
Were you able to quantify if there's anything special about Manuka honey over West Australian honey?
That's a little bit tricky because my PhD is mainly focused on the Western Australian honey. So especially Western Australia doesn't produce a lot of Manuka honey. And it's really hard to get the honey from outside of Western Australia. So I haven't been analyzing quite a lot of Manuka honey in terms of that authentic Manuka honey. So I can't explicitly make a comment on that. But so far my understanding is that the Western Australia, due to its unique geography, it has a really good potential, especially its iconic honey, Jarrah honey, then Mary honey, some of the honey that we have already identified. That was not on the focus yet for the industry, but that has really good activity, especially in the lab setup. So if the industry wants to capitalize that one, they can also focus on that particular honey as a, how can I say, to promote that, to build a really big market for that honey store.
Yes, please.
Yeah, excellent presentation, well done. Contamination is a big issue in honey.
Yeah.
Do you think there is a scope of your methodology to fingerprint contaminants in honey?
Actually, the contamination, if you think about contamination, there are two possibilities of contamination, especially one is for environmental prospects, like the trace element or even pesticide herbicides. And the other aspect is the fraudulent activities. Some people added something. So in my PhD, I mainly focused on what the people are adding into the honeybee product, especially the cheap sugar syrup. So we can pick up the cheap sugar syrup, especially if someone added in a really high quantity, that is make them profitable to mix that. Otherwise, no one is going to add 1% or 2% just to get caught. So we can pick that, especially the fraudulent activity. In my PhD, I haven't focused on the environmental contaminants, so I can't make any comment on that.
I might just add one question, if I may.
Yeah.
Obviously, there's a lot of industry application for this kind of work. How can a farmer access these sorts of analysis? Is it about yet? Or?
Yeah, one of the good things is that as my PhD is funded by the Cooperative Research Center for Honeybee Product, and we find out that, okay, there are a lot of potential for the method that I have already developed. Because I have already mentioned in my presentation that Kamag has already adopted one of their methods. CRC has already promoted the fingerprinting tools that we are using as a label. So we have also presented that in the beginning of this month in the bee conference in Sydney. So that has also grabbed a lot of attention. But as a beekeeper, how can they access that? So there are not a single honey lab in Australian context yet, but due to the interest for the beekeeping industry and from the beekeeper, CRC has already a plan, and they have gone a green signal that, okay, they are going to set up a lab in Yanchep, especially it will be closer for the WA and also for the WA beekeeper. And I hope that will be running with the end of this year. And so that the beekeeper can access all of these analytical techniques with a really cost-effective manner.
That's great news. Okay, we have one more question.
Okay, wonderful talk, very interesting, very informative. Honey is the product of the interaction of the nectar and those things from the plant side and also interaction with the bee contribution to the honey. Do you think it's worth looking at bees as well, whether different bees produce different type of binding patterns? Whether have you done this type of work? If not, is that worth doing? Thank you.
Thank you for the question, because I actually expected this type of question. One of the interesting aspects is that the honeybee so far is harvested for the commercial quantities from the European honeybees, that's Apis mellifera. But Australia has a quite large number of native bees, and quite a few also produce enough honey that is that can be, or that has already available in the market. So that's also a good thing that my supervisor has already initiated a master project, and we have already get a master's student, our lovely Mariana from Brazil. She's going to look into all of the, especially the Australian native honeybees honey. We have already received quite a few number, about 20 of the honey from New South Wales and also from Victoria. We're expecting to get quite a more honey along the way, especially the next 2 years. So we hope we'll find the answer of your question, because as we haven't analyzed the native honeybee honeys yet. Thank you.
Great. Thank you, Khairul. That was a fantastic talk on such an important industry issue. You did a fantastic job.
Thank you.
Our next presenter is Tanushree Halder from the UWA School of Agriculture and Environment. Tanushree first became highly interested in crop improvement through exploring plant genetics and breeding during her Bachelor of Science in Agriculture at Sherlabunga Agricultural University in Bangladesh. She was awarded a gold medal and the Dean's Award for her outstanding academic achievements in her bachelor's degree. Furthermore, she successfully completed a master's in genetics and plant breeding from the same university and joined as a faculty member. Prior to joining her current PhD program at UWA, she's completed another postgraduate degree with a Master's in Applied Biotechnology from the University of Westminster in the UK. Her PhD research is supported by the UWA RTP Scholarship to investigate the genomic regions, genes and proteins associated with different root traits in wheat. She has recently published one research paper and a review from her PhD. Very impressive. Tanushree's presentation today is entitled Unraveling the Genetics of Wheat Root System Architecture. Please join me in welcoming Tanushree.
Thanks, Hayley, for the nice introduction, and good afternoon, everyone. We all know that wheat is an important economic crop which contributes to 68% to food and 20% to feed in the world. So today I will talk about that why we need to improve wheat production and how my research contributes to that. Here are the key people and the funding bodies who are supporting my research, and I'm cordially thankful to them. Now coming to my first question, why we need to improve wheat production? Currently 776.8 million tons of wheat are producing in the world, and Australia is contributing 36 million tons among it, and it's exporting 70 to 80% of it. However, by 2050, it has been predicted that the world population will reach to 10 billion, and we need to fill up the gap of wheat production, 56 million tons. Now, what are the current challenges of wheat production? We all are aware about the climate change, and among the climate changing options, the major constraints are drought, salinity, waterlogging, and heat. Now, we believe that at this condition, an improved root system of wheat can help us to develop the climate resilient as well as high yield potential wheat varieties. Why? Because root is the key role player to supply water and nutrients to the plants at any environmental condition. So if we can improve the wheat root system and can ensure the continuous supply of adequate water and nutrients, only then wheat can survive and can produce higher yield through the photosynthesis. Now, how can we improve the root system genetically? There are two options. One is conventional breeding, another one is molecular breeding or furthermore genetic engineering. However, over the conventional breeding, molecular breeding allows us to improve any specific root traits in a shorter time and with high genetic gains. Therefore, we are focusing on molecular breeding. And here, my research objectives are to identify and validate quantitative trait loci for root traits of bread wheat, to identify candidate genes and proteins which are associated with the root traits of wheat. In my experiment, I have used two different pure lines, recombinant inbreed lines or RILs and near isogenic lines or NILs. We used the pure lines because it has the stable genetic constitutions. So in any environmental conditions, we can grow it and multiply it without changing its genetical constitutions. And recombinant inbreed line has been developed by crossing synthetic and opata and through the advancement of 8th generation, where near isogenic lines are the isolines, or we may say pairs, which has the common genetic backgrounds but has only one contrasting genomic region that control that any specific trait. Here we can say that if we find any kind of variation for the trait, that is controlled by that genomic region. And as they are pure line, they are helping to get the reliable genetic data of any trait. We have grown the wheat plants in a semi-hydroponic system, which is very good growing system to study the intact root system, growing larger number of plants in a smaller area and having the homogeneous environment, which is not possible in case of soil, because if we want to study root system from the soil, we need to invade the soil, we will lose the root, and also it will disturb the soil structure. Therefore, it is an excellent system to grow plants and study roots. And after harvesting the plant, we cut the root and then scanned it through a scanner and further analyzed the root using the WinRhizo software. Having the phenotyping data, we furthermore analyzed it for the genetic study. We identified QTL using QTL mapping, and we used RILs as a population, and identified candidate genes using genotype-phenotype association analysis and proteins using proteomic analysis, and as a material we used near isogenic lines. I have studied 12 important root traits for my study, and today I will discuss about mostly rooting depth, total root length, root mass, and a little bit of root diameter and root surface area, which is more than 0.25 millimeter diameter class, commonly known as coarse root, and another surface area which is less than 0.25 millimeter in diameter class, which is commonly as fine root. Here is an example of two contrasting genotypes. Now, first of all, we need to know what is rooting depth. Rooting depth is the length of a root that enters into the highest depth of any growing system. So here we can see that genotype A has longer root, 75.5 centimeter, B has shorter root, 55 centimeter. However, by looking at this only contrasting phenotype, we can't say that, okay, the total root length is also like 75.5 centimeter because there are another roots. So here comes the total root length which has been generated from the scanning. And here is the data of genotype A and genotype B. Looking at this table, you can see that genotype A has higher rooting depth, total root length, root mass, as well as fine roots is higher. However, in case of B, they have more coarse root and the diameter is same. But looking at this table, can we say that we should select only genotype A for having a climate resilient and higher potential wheat genotype or wheat variety? No, because in different climatic condition and different soil condition needs different types of root traits. As an example, in case of drought, when the soil is loose and the water level is very low, then we need deeper root length, more deeper rooting depth, more root length, root mass to uptake the water and nutrients. However, when it is well-watered condition, if we have more total root length and root mass, the energy goes there and yield can be lower. On the other hand, in compact soil where the water level and nutrient level are close to the top soil, we need shorter root system so that they can uptake the water and nutrients and contribute to the yield. And also for the fine root, we need more fine root for uptaking water and nutrients, and we need also more coarse root to have a good anchorage. So we can see that we need different kinds of root traits for different climatic condition and different kinds of stress, and also depending on the region. And therefore, we need to know genetics of single traits, not means many traits, but individually, to know the combination as well as their individual role. In my experiment, I have found a lot of variation in rooting depth in case of recombinant inbreed lines. You can see that highest is the 88 centimeter and lowest is 20 centimeter. I have also found significant difference between the isolines of different nil lines. As for example, nil pair 6 and nil pair 9 and 10 showed significant difference in root length. And I need to mention here that the nil pair 9 was developed targeting the chromosome, developed the genetic region targeting the chromosome 4A, and nil pair 9 and 10 was developed targeting the genomic region on chromosome 7A. Nil pair 6 also showed significant difference for root dry mass. Now, how the phenotypes is connected with the genetic constitutions. We know that every trait is the expression of protein, and this protein is encoded by the gene, and the genes are located on the DNA, or in a large term, chromosome. So if we know that every information, then we can improve the specific traits. As for example, if we want to improve rooting depth, then we need to know what are the key genetic factors that are controlling the rooting depth. And hence, my first research objective is to identify the quantitative trait loci for the wheat root traits. As a definition, the quantitative trait loci is the genomic region where the genes for any quantitative trait are located and control that trait. And I will explain it later in my next slides. And we identified 22 QTL altogether, and except few chromosomes I found in different chromosomes and different QTL. Before going to the actual picture of QTL, I want to show that wheat genome is a complex genome which has 3 different genomes, A, B, D. And these 3 genomes has 7 copies of chromosome, which is very complex. And today, I will give an example of QTL on chromosome 5A, 5D, and 6A. Now coming to the example. In this chromosome 5A, in this region, I found the QTL for total root length. Here, the QTL for rooting depth. In chromosome 5D, in this region, QTL for, again, rooting depth and fine roots or root surface area, which is less than 0.25 millimeter in diameter class. And in 6A, in this area, this is the QTL for root dry mass. From these example, we can see that any trait is not controlled by different trait is not controlled by only one chromosome or only one region. They are distributed in different chromosomes and different genomic region. Therefore, these 22 QTL that we identified for root traits are important for further research. Now coming to my second objective, that which candidate genes are responsible to control these kind of root traits. I identified 15 putative candidate genes which control different root traits in near isogenic lines. Now what's novel? I found 3 novel genes which control wheat root traits and which encode 2 different proteins. One is UDP glycotransferase, or in shortly UGT, leucine-rich repeat receptor like protein kinase, or in short, LRR-RLK. That means they were identified in other plants, as for example rice, canola and Arabidopsis, but we reported it for the first time in wheat. Now what is the pathway? From the literature, we found that UGT contribute to root growth and development through glycosylation of 3 important phytohormones, auxin, cytokinin and abscisic acid, and also contributes in lignification. On the other hand, the LRR-RLK contributes to root growth and development through cytokinin mediation and stem cell signal transduction. Now, the third objective, we have already identified the QTL, candidate genes. Now, what are the proteins? Analyzing the Neal pairs, I identified 114 differential expressed proteins in Neal pair 6. So, you can see these are the differential expressed proteins and these are the common proteins. And here is the example of 4 important proteins. If we notice here, the UDP glycotransferase that we have identified already as a novel protein through our candidate gene identification, it is here also. And the other proteins are cytochrome B561 and domain containing protein, asparagine synthetase and thyrodoxine. Again, from Neal pair 9, we identified 30 differential expressed protein, and here is the example of 4 important proteins. Also, you can notice that this protein is the other novel one, leucine-rich repeat receptor like protein kinase, 60S ribosomal protein, signal recognition particle protein, and phosphate synthase. So, with these 3 experiments, are there any common factors that are controlling the root rates? Yes. After the QTL analysis, I have found that the QTL which is controlling root mass in 6A, they have a gene which encode UGT. Also from candidate gene identification and protein, I have already demonstrated that we found UGT. So this is the common one. And if you can remember that the UGT was identified in Neal pair 6, which was developed targeting the chromosome 4A. So we can say that chromosome 6A and 4A has a gene which encode UGT, and from the candidate gene and protein identification we found UGT and LRR RLK protein similar. In a conclusion, so in my research I have found a lot of phenotypic variation from reals and Neals, and then we identified 22 QTL which is controlling different root rates, 15 putative candidate genes, and among them 2 were novel, and then in total 144 differential expressed protein which has commonly the novel proteins again, and we have validated few QTL, and currently we are validating the genes and proteins that are controlling the root rates. Then what next? My research outcome can contribute to the improved root system because these molecular marker and the proteins information we have identified that can be used in further breeding, molecular breeding, as for example, marker assisted breeding, genomic selection, or further genetic engineering, as for example, CRISPR-Cas. So my research is contributing to the improvement of wheat root system and ultimately to develop the climate resilient and high yielding wheat varieties. Thank you so much.
Wow, things have changed since I grew a few clovers in a paddock at Shenton Park, haven't they? Fantastic talk, Tanushree. Thank you.
Thank you.
Do we have any questions?
Thanks for a wonderful talk. Very interesting the fact that you found candidate genes common across species. Does that potentially mean in the future we might even think of diagnostic markers that actually work across species for modifying crop root systems?
Thank you for the question. What I understood from the question that if we can use this marker for further investigation?
Yes, and it seems to me I'm there's amount of commonality between across species that in fact it will be much more applicable to just wheat, and you will be able to potentially use even transgenic sources of root architecture from other species within wheat.
Yes, thank you so much for the question. Now I understand. Definitely,
Thank
as
you.
it is from pure line, so we are expecting that we can use these molecular markers for other species to improve their root system.
Great. I think we have.
Yes.
All right. Sorry, John. Just one first, Jackie. Sorry.
Thanks for an interesting talk. You've got 22 QTL region. How did you identify the 15 candidate genes from within that?
Thank you very much for the question. But we have identified the candidate genes from near isogenic lines, not from the recombinant inbreed lines. We identified the QTL from the recombinant inbreed lines and candidate genes from near isogenic lines.
But how did you get down to those 15 candidates?
Candidate genes using the wheat 90K array analysis. So we have done the SNP analysis. We had the phenotypic data and had the SNP data, and we analyzed that with the genotype-phenotype association.
Yeah, so you presumably got a genomic region. So how did you pick those candidate genes within that genomic region? Was it purely based on the fact they'd been identified in other species, or were they the only 15 genes within your genomic region associated?
No, that 15 is within the genomic region.
So they're the only genes within that region?
Yes.
Okay.
That 15 genes. Do
And
you want to see that?
No. Just because. And a follow-up question,
Yes.
how are you validating them?
I want to validate them using the RT-PCR.
Oh, so you're just going to look at expression?
Yes.
Okay.
Yeah.
So have you looked to see if there's any sequence difference in them at the moment?
No, I didn't see the sequence differences yet because I'm now designing that. So I'm on the process of this.
Sorry,
Thank
just
you,
interested.
Vicky.
Thank you.
Thank you.
Fantastic. Still got a question? Oh, sorry, I missed somebody. Oh, yeah, sorry. Get back to you. Can you speak louder? Oh, yeah, I'm sure you can speak loudly. Thank you.
Thank you, Rojana. Yeah.
Yeah.
So very nice work, Tanushree. And I think this is really good that you try to correlate different things. Two things. One is just follow up what Jackie was saying. So I think will be good to go in the deeper in the QTL region with the genome sequence and then try to see what kind of genes are there and whether some of these genes they are also present in your gene expression analysis. Okay, so you can do the bioinformatic analysis in QTL region and try to see. So this is point one. Second is that this population, the RIL, this is the synthetic wheat population,
Yes.
which is good. It will be good idea to validate some of these QTLs on some population which are derived from the drought tolerant or drought susceptible genotypes or the different root architecture. So I think this will add value further for your work.
Yeah. Thank you, Professor. And we have done actually some validation of the QTL, and that I told that for the root mass one, I have validated them in another population which is Opata and Cascade. That was validated already. And for the gene synthesis analysis, the QTL that I identified, I tried to identify some genes from the QTL region, and that actually showing that the UGT is also there. Very good. Thank you.
One last question. Thank you.
Fascinating talk.
Thank
But
you.
the problem I see is that your screening system was done in a water culture, and in reality, they're going to be having to cope with a whole range of different soil types.
Yes.
How do you see it, the technology transferring into the real world?
Thank you for the question. I used this system, this semi-hydroponic system, but previously it has been already validated from other PhD student who has studied this kind of root system in the rhizobox. Means previously it was studied in semi-hydroponic system, and then they repeated it in the soil system, and they found that there is no significant variation. So we can expect that it will behave the same. But definitely in different climatic region, then we need to think. But as they are pure line, they should behave similar.
Great. And we have one more question.
Thank you.
Thank you.
I'm very confident. Well done.
Thank you.
I'm not a gene jockey, so I don't know anything about genes. I'm a soil scientist down here. You started your presentation about climate change, some of the issues like salinity, drought, heat. So my question is, some of these abiotic stress, do you think they will have any impact on this molecular marker or protein marker?
Good question, Professor. Thanks. As this is from pure line, we are extracting this marker. We are expecting that it should behave same in different environment. However, as I already mentioned, that different soil structure may have the role for root plasticity definitely. So that is for future work. Definitely, we need to go further. We need to interrogate this marker, and then we can validate that. But from the hypothetical things, it should work. That's the thing.
Thank you. So I think you'll agree that was another fabulous presentation about a subject that's really important to our industry and important to the future of our wheat growing industry. So please join me in thanking Tanushree.
Thank you, Hayley. Thank you so much. Oh, thank you. Mr. Thanks.
I have the pleasure of introducing our final presenter for this first session. So Joe Gebbels is from the University of WA School of Agriculture and Environment. Originally from the UK, Joe first came to Australia in 2010 for a uni-prac and went down to Hyden or Lake King and spent a year on a farm. And I'm sure after being in the UK, Hyden, Lake King must have been a bit of a shock to his system. Since permanently relocating to Australia in 2016, Joe has worked for Meat and Livestock Australia as the Program Manager for Sheep and Goat Research and Development, first based in Sydney and since 2019 based in Perth. In addition to his role for MLA and wrangling a toddler, Joe has been undertaking a PhD focused on investigating strategies for reducing emissions and building climate resilience in agriculture in Western Australia. This afternoon, Joe will be exploring his research into improving productivity and reducing methane intensity while increasing, let me try that one again. It's too many words here, Joe. Improving productivity reduces methane intensity but increases the net emissions of sheep meat and wool enterprises. Please welcome me in joining in. Welcome, Joe.
Thank you, Hayley. I should have shortened it, shouldn't I? So the focus of my PhD really is on reducing agricultural emissions in Western Australia. And today I want to talk to you about a particular paper that I've just finished with the support of my supervisors that's looking at the methane intensity of Western Australian sheep meat and wool. Before I go into the specifics of the particular paper, I just wanted to give you a bit of an overview of agricultural emissions in Australia. So if we look at the economy as a whole, agriculture is responsible for around about 13% of total national emissions. Of that 13%, about 80%, roughly 10% of the total, is attributable to the production of ruminant livestock and the release of methane through the process of enteric fermentation, the digestive process by which ruminants break down fibrous materials in their diet. There are two ways that we can look at emissions, and I'm going to focus on both of these metrics as we go through the talk today. The first is to look at the emissions, or in this case, methane intensity of a product, which is quite simply the kilograms of emissions attributable to the production of a kilogram of product. And it's a way for us to compare a range of different products on a unit basis. It could be lamb, could be avocados, could be essentially any product that you like. And then the second opportunity to look at emissions is to look at net emissions, which is just a way of looking at total emissions and enables us to compare different enterprises and different sectors. So I'm going to use both of these metrics as we go through the talk today. I think the challenge more broadly for the agricultural sector is that because of that dominance of methane within the agricultural emissions profile, reducing agricultural emissions is very difficult. And unlike, for example, the energy sector, at this stage, we still don't have a lot in terms of market-ready mechanisms that can permanently reduce agricultural emissions. So I think there's a real need to optimise the efficiency of our current systems whilst continuing to explore what some of the transformative options are in the longer term. So that brings me on to the key aims of this project that I wanted to talk to you about today. I wanted to look at what are the drivers of variation in methane intensity of sheep meat and wool produced in WA. I wanted to look at the change over time. So we took a base year, which is in this instance is 2005, and looked at the change through to 2019. And then we also wanted to look at a series of future scenarios out to 2030, but also thinking about what are the limits of our current production systems and how far can we optimise the system. So why do we want to do that? Well, I'm sure you're all aware, particularly having gone through last summer in Perth, we've seen a significant warming trajectory since the pre-industrial era. And if we look out to my now 2-year-old son's 80th birthday in 2100, he's potentially looking at as much as 6-degree rise in mean global temperatures. So the red line is based on our current emissions trajectories, and then the yellow, blue and green lines are at various different levels of emission reductions. So I think there's some pretty sobering, that's a pretty sobering thought, particularly if we think about our existing production systems. The red star there is where the CSIRO says we are today, so we're already tracking above the higher level forecast. And that obviously is going to be a big challenge for agricultural systems anyway. We're forecasting an increase in extreme events, things like more frequent and more prolonged droughts that are going to really be a challenge for our producers. There's also the issue of policy and public perception, and I think there's been a general growing awareness and recognition of the role of agricultural emissions and the importance of them. And if we look at the household level, about 15 to 29% of emissions attributable to a household are directly due to the purchase of food. If we look at what the governments around the world have been doing to try and reduce agricultural emissions, they've typically, certainly in Australia, taken a beneficiaries pays approach. They try to incentivise producers through a variety of mechanisms to reduce emissions. But I think we're seeing a gradual transition in the narrative towards more of a polluter pays based approach. And if we look over at New Zealand, they've actually legislated reductions in agricultural methane emissions by 2050, and more recently they've actually announced that they're going to introduce a price of 11 cents a kilo for methane emissions, which by my calculation works out at roughly 70 cents per year per sheep. So there's definitely a shift in that narrative there. I think from an Australian and a West Australian sheep industry perspective, really what we want is a sustainable operating environment. We want to reduce the risk of policy interventions that might make our production systems more challenging, and we also want to ensure that our production systems are profitable and efficient as possible. So we, to start our project, we undertook a comprehensive literature review to identify some of the key drivers of variation in methane intensity, and we came up with the variables you can see on the right-hand side there. So I want to draw your attention in particular to marking rate, which is quite simply the number of lambs produced per ewe. So if we have an 80% marking rate, that means that 100 ewes produces 80 lambs on average. And also carcass weight, which I think is fairly self-explanatory, the average weight of a lamb carcass. Range of other variables that we looked at there, including the proportion of ewe lambs that get pregnant within their first year, sheep death rate, growth rates, ewe standard reference weight, which is just in essence how heavy the average sheep is, ewe longevity, and also flock composition. I did just want to mention that our focus is very much on the productivity drivers of variation, and other areas of research, like research into anti-methanogenic compounds, are not within the scope of our current study. So what we did is we mined a series of industry data sets and tried to identify a baseline level for each individual variable in 2005. So for marking rate, that was around about 80%, and for lamb carcass weights, it was around 21 kilos. We then, with those same data sets, looked at the change over time and through to 2019, so we could see what changes already occurred, and for marking rate, that takes us up close to 93%, and for carcass weight, close to 23 kilos. But we also wanted to look at future scenarios and what might happen in the future. So we developed 3 scenarios to do this. One was just linear extrapolation for each of the variables. So whatever the change was between 2005 2019. We just carried that through on a linear basis to 2030. And then for our second two scenarios, we did an expert consultation process where we asked some leading producers, researchers and consultants what they thought would happen out to 2030, but we also asked them what they thought the limit was under our current production systems without a time-bound requirement. So this looks a bit scary, but that gave us a complete data set with our base scenario 2005, 2019, linear extrapolation, 2030, and then practical limits. So again, I'm just going to focus on marking rate, which under linear extrapolation I'll see is at close to 102% by 2030, which interestingly was quite similar to what our experts thought would happen. And then the practical limits was considered to be around 146%. So 100 ewes can potentially produce 146 lambs under our current production systems, is what that process said. And similarly with carcass weights, we see quite a general increase in forecast carcass weights up to a limit of close to 28 kilos. So we took all of this information and we put it into GrassGrow, which is a livestock systems modelling software package, and we simulated two representative sheep enterprises. The first of which was at Pingelly, where the UWA future farm is. Pingelly is a medium rainfall environment in the sheep wheat zone in WA, and our second site was out at Merredin, which is a medium to low rainfall environment in the eastern wheat belt. The enterprises that we simulated were the same in both situations, a self-replacing merino flock with a small wether flock as well, and we set a series of constant assumptions around supplementary feeding strategies and also stocking rates, so that the scenarios that we were comparing were consistent over time. The results of that process show us that from a methane intensity perspective, so on a kilo basis, our emissions are going to be 19% lower in 2030 than they were in 2005. And if we look at our expert scenarios, we see that reductions of up to 42% are considered possible under our current production systems. So if we look at the drivers of that change over that period, we can see, as I've already said a couple of times, that marking rate and carcass weights are really the two biggest drivers. And essentially what that's doing is producing more kilos of lamb per ewe and diluting her emissions over those kilos. So the more that she can produce, the more efficient we are. Other variables that had a positive effect on reducing methane intensity were ewe death rate, ewe longevity, so keeping them a bit longer, increasing ewe lamb mating and reducing the wether proportion of the flock. When we look at growth rates, it had relatively little difference in most of our scenarios, and it sort of depends on whether it reduces our time to slaughter or not. And the only variable that actually increased methane intensity over the period 2005 to 19 was increases in ewe standard reference weight, or just weight, which had a minor increase. So we've got slightly bigger ewes than we did back in 2005. This is just a representation of the impact of a 1% change in any of these variables on methane emissions. So if we change carcass weight or marking rate, which is pretty much the same, by 1%, we see about a 0.5% change in methane intensity as a result. And you can see that the other variables, whilst they're having some effect, is quite muted in comparison. I think this is the challenging other side of the story, which is that as we bring methane intensity down and our flock productivity up, we see an increase in net emissions, which are forecast to be 22% higher in 2030 than in 2005, unless we also constrain enterprise scale. And in our future scenarios, that means that there's potentially an increase of up to 61% in net emissions. Why is that? Well, 87% of the variation in methane emissions is due to variation in feed intake. And we've got a series of things within our production systems which are driving the need for increased feed intake. We've got more lambs that we need to feed. We've got ewes who have a higher energy demand due to that increased gestational and lactational energy requirements. And we have slightly bigger ewes as well. So that more feed translates pretty directly to more emissions. So in summary, we're producing a lot more product per kilo of emissions than we were in 2005, and we still have quite a long way that we can go in terms of reducing that further on a kilo basis. The real driver of that in a flock is improved reproductive performance and diluting that ewe's emissions as much as possible. But if we want to reduce net emissions, we'd also need to see a reduction in ewe numbers to prevent that increase in feed demand offsetting the gains that we get from an intensity perspective. So productivity in itself is not directly a pathway to reduced methane emissions within a sheep enterprise. And I think if we sort of think about the implications of that, and it's probably why, in my view, there's been relatively little in terms of direct policy in this space, is the link between higher productivity and higher emissions makes it quite difficult to sort of incentivise emissions reductions directly. But I think from a producer perspective, it's actually quite a bit simpler. It doesn't matter if somebody's going to incentivise you to reduce emissions, potentially through a premium, if they're going to cap your emissions, or if they're going to set a price on your emissions. The more efficient you are as a producer, the better off you're going to be under any of those scenarios. And from a research perspective, and you know, important for many of you in the room, we're going to have to look at other pathways if we're going to reduce net emissions. So I'll finish up there, and happy to take any questions from the floor. Thank you.
Great. Do we have any questions for Joe? Oh, sorry, John Hamlin.
What do you see the role of societal change in terms of meat consumption affecting this type of issue?
I think that there are probably instances where consumer preferences are shifted by that growing awareness of the emissions profile of meat versus alternatives. But I think when you look at the global scale and in the context of a growing population, I think that the just overall demand for protein is probably going to dwarf the impact of individual consumer choices in particularly developed countries.
Cool, thanks Joe. Really interesting. One of the questions I've got is around the pasture base and kind of what the model, pasture base, the model was based on, and just how you see potentially any change with different pasture mixes that could be used.
As I interpret the question, I sort of interpret it thinking if you get a better pasture base, what then happens to the emissions profile? And I think the answer to that is if you get a better pasture base so that you can produce more kilos of animal, probably the producer will also expand the flock in order to utilise that resource as effectively as possible. So you're going to bring your intensity down by improving that, but the commercial driver at the moment is just to optimise your stocking rate and therefore probably expand your flock size.
Joe, interesting presentation, thank you. The intensity of the emissions, were you able to get any relationship between the higher intensity and the better productivity through the utilisation of methane within the animal?
Sorry, could you repeat that question?
Yep. As the intensity of your methane increased, did you explore whether there was any positive impact upon the energy utilisation of methane within the animal as a result of that higher intensity?
So in each of our scenarios, the intensity decreased, the methane intensity on a kilo.
Yeah, as the intensity increases, then it may have to be then coming back to how you reduce the methane metabolism within the animal with bromoform and so on.
Yeah.
So I was just wondering whether you explored whether there was a productivity outcome which is beneficial to the producer, because I think most of your measures of reducing it were farmer measures to adapt.
Yeah.
I was just wondering whether there was a bit of a bonus somewhere.
Yeah, absolutely. So to answer the first part of the question, Grassgrow, the software we use, applies a static assumption based on feed intake for methane output. So we weren't able to look at potential for anti-methanogenic compounds, for example, to play a role, but it's a really active area of research, and that's why I tried to highlight that it was separate, and I think it's probably to the scope of our current study, but I think it's one of the key areas of research that really needs to be focused on in terms of direct reduction because of the challenge. Otherwise, we'll see an increase as productivity improves.
I'm sure Phil would agree with that.
Joe, very interesting talk, thank you. Interesting that New Zealand are putting a price on methane. Obviously, more lambs plus heavier lambs equals more money for the grower. Did you have a look at what price methane would would have to be costed at to bring everything back to square? In other words, how much could the grower afford to pay in a price for methane?
Yeah, I think it's a difficult one because it's tied into consumer willingness to pay for a product. I think what the New Zealanders have tried to do is bring in, what is probably still a relatively modest emissions price at this stage, and my understanding is that funding will be diverted into research to try and develop other pathways to reduce emissions. I think this is one of the real challenges that we have as an agricultural sector. With the energy sector that has these mature technologies like solar and wind, if you increase the price of emissions over time, you just push them further towards those market-ready mechanisms. You sort of reach a tipping point. I think the danger for agriculture is we don't have those commercially available options at this stage, and so you're in effect just penalising industry, and it hasn't got anywhere to go at this stage. So I think what the New Zealanders have done in terms of investing in research is probably a pretty sensible strategy from my view.
Great. One more question.
Great presentation. Unlike Phil, I'm not responsible for methane. I'm more responsible for nitrous oxide and soil scientist. You see Australia, food security may not be an issue for Australia, but we provide food security for many nations. So in your opinion, is the intensity is the issue or net emission? Because we want to increase the food production, meat crop production. What is the government policy? Is more into net emission or intensity?
I think you have to look at net emissions at the whole economy basis. It's obviously very clear that we need to reduce emissions, and I think there are probably easier short-term targets than agriculture. I think, though, if you were to set a cap on agricultural emissions and say this is your limit, might be whatever the current level is, we've still got a lot of room within our production systems to become more efficient and to produce more product within that potential cap. So I think it has to be both ultimately. But yeah, I think when you look more generally, there's probably lower hanging fruit in terms of immediate net emissions reductions.
Thank you, Joe. Another insightful presentation, and I think that's one of the major challenges facing our meat production industry in Australia. So thank you. Join me in thanking Joe.
Thanks very much.
Thank you, Joe. Hayley, don't go.
Before I go to the toilet, haven't I?
Yes. Thank you, well done. So please join me in thanking Hayley for the wonderful chairing. And also the other three speakers. This is thank you. And Joe, I enjoyed your talk as well. This makes me compelling evidence that we should control the number of sheep on UWA farm. So from 8,000, we are thinking of around 4,500. My animal scientist won't agree with me that. Again, wonderful talk. So we're going into the next part. So we have now Professor Ann Novak here. I just want to introduce her. She had some other engagement, so she could not come at the beginning. Anna, this is our 16th annual postgraduate showcase, for your information. And many of the audience here represent various organizations, Murdoch, CSIRO, DPIRD, farmers, industry, and so on. So some of them are regular because we have a drink at the end. It's now my pleasure to introduce UWA Deputy Vice Chancellor Research, Professor Ann Novak. Anna has responsibility for research policy development, leadership of the university's research activities, postgraduate education, industry liaison, intellectual property, commercialization. You heard from some of the first talk, things are getting the PhD into commercialization mode in the honeybee industry. Anna is a medical oncologist, clinician scientist, so she's not supposed to work on Fridays. Go and do your clinical job. Completed her undergraduate in medical degree and her PhD in tumor immunology from UWA, so a UWA product. In 2018, Anna was named the Cancer Researcher of the Year by the Cancer Council of Western Australia, and in 2021 received both International Mesothelioma Interest Group's prestigious Wagner Medal for her contribution to mesothelioma, and Research and Medical Oncology Group of Australia's inaugural Martin H. Tetzel Heroes Award for mentorship, advocacy, and contribution to the discipline. Anna's research leadership experience includes her role as the Pro Vice Chancellor, this is the previous role, Health and Medical Research at UWA, Associate Dean Research of UWA's Faculty of Health and Medical Sciences, as the Director of the National Center for Asbestos-Related Disease. You're still holding that position since 2018, and the Chair of the National Brain Cancer Collaborative Clinical Trial Group, COGNO, from 2017 to 2020, and Anna is also a member of the Cancer Council of WA Board. We are very thankful for you to come. I know you had a number of other engagements, but it's great to see you here. Anna.
Thank you, and I'm probably going to be a disappointment after that glorious welcome. So look, it really is lovely to be here and to see you doing for your HDR students what we should be doing across the whole university for our graduate research students, which is giving them the opportunity to present their work and to network and to get out there and work together with industry and other universities and their academic colleagues. So this morning, I've come from the University Research Committee Strategic Planning Day. And really, many of the things that we aspire to do in the university are actually exemplified by what is being done in the Institute of Agriculture. So when we're looking at building research capacity, we're really looking at what is the secret sauce that's going to make us successful as a university, and also what is success. And when we look at the different ways of measuring success, we can look at world rankings. We've got great success in agriculture here at UWA. We can look at industry engagement. It's something that the Institute and the schools associated with the Institute do incredibly well. We can look at highly cited authors, also something that has grown out of the Institute of Agriculture with a number of extremely well-regarded colleagues in this room. And we can look at the funding that is brought in for research. So on any of these metrics, the Institute for Agriculture clearly demonstrates a critical mass that is really accelerating not only the university's trajectory, but more importantly, the discipline and the benefit that we're seeing to the world out of the work that you do. So some of the themes that came out of today's University Research Committee planning meeting, we were looking at what are the overarching themes that a number of schools can contribute to. And these included sustainability, food security, social impact, big data, climate change. And these are all things that you're already doing through bringing together people from a diverse set of disciplines and working in a transdisciplinary and multidisciplinary way to do that. And so I thank you for your open minds and for your collaboration as you bring together agriculture and economics, agriculture and health, and engineering agriculture, and so many other cross-disciplinary pieces of work. One of the other themes that came out of this morning's workshop as well was the importance of end-user engagement, and that we should be starting as researchers in a university community as a whole to talk to the people for whom our research is important before we even design our research. And so that really close relationship between the grower groups and industry, between government and DPIRD and the Institute and the university is absolutely critical in ensuring that the most impactful research is the research that's being done. And we don't do that so well in many other areas of the university. along with fostering partnerships and certainly evaluating economically what the value of our research is to our community. So on Tuesday, we've got an all-staff forum coming up, and I'm sure that you've all heard from the Vice-Chancellor over the last couple of years that our priorities are around student enrolment, student experience, and financial sustainability. But it is impossible, I think, to untangle any of those from our research excellence and our research impact, because students want to come to study at a university that is doing excellent research, that is building the reputation, that has researchers and who are also excellent teachers and who are well regarded and internationally connected in their discipline. Our financial sustainability also relies on research income, of course, so we just can't unpick these at all. So I'm really delighted to see that our Vice-Chancellor is adding, and our Senate, building research capacity as one of the 4 pillars that we will be talking about strategically within the university. So all of those who live for and love research, we can all start to feel that research is going to be back on the agenda, and visibly so in UWA. And along with that, the importance, of course, of HDR, PhD students. You are our pipeline, you're the next generation. You're going to be sitting in these chairs in a number of years' time, we hope. So that's really all I wanted to say. I thank you for all the work that you do under the Director, as collaborators, as students, and as people who are engaged with the Institute of Agriculture.
Thank you, Anna, and you've spoken so well. Unfortunately, I didn't bring a wine because you gave a lot of praise for the agriculture part of this university. Certainly, we thank you for that. And also, it's pleasing to see that a research-intensive university, such as UWA, will enforce again one of the major traits in this university. So that's really good. And certainly, the PhD is one of our strengths in agriculture. The undergraduate numbers are not that large, which we admit that, and it's not going to change dramatically, but the masters, particularly with international students. But the PhD is very strong. And the beauty is that a lot of those PhDs, or almost all of them, have implications for the industry. So we thank you. Hope you can stay here. Now, for the audience, we have a tea, 2:40. In fact, we are early to 3:20 p.m. in the foyer there. So there is a coffee, tea, and some bickies, and refresh yourself. And you'll be back here at 3:20. And then the speakers, the students, Anna, before you go, and also Graham, who is the trainer of the students. We want to get a photograph somewhere outside. So enjoy, and we'll see you at 3:20. Thank you. Okay. Thank you everyone. Welcome back. I hope you had a bit of a networking and refresh yourself, and we got 3 more presentations this afternoon. It's my pleasure to introduce the chair of the second session, Emerita Professor Lynn Abbott. Lynn is a world-leading academic and science communication in soil science, soil biology at the UWA School of Agriculture Environment, and of course within the Institute of Agriculture. Lynn has had many roles in the university, but more importantly, she has conducted pioneering research in symbiotic soil fungi in agriculture and disturbed natural environments during an academic career that has spanned more than 5 decades. So almost as much as Graham. Professor Abbott established the innovative Know Your Soil Biology workshops for farmers and provides hands-on activities demonstrating the benefit of soil biota, soil health, especially for the school children. She has done a lot of work with high school students when we talk about STEM and encouraging students comes to science and in agriculture. And she leads the land restoration demonstration site research project at the UWA farm. Some of you may have seen one of the most difficult landscape in terms of water logging. No one wants to take trial, but Lynn ventured to that. Even first time when she took me through a ditch to reach there. But when we went and looked at 2 years ago, it's grown so much, and a lot of students have worked. Enormous amount of data has been collected. Lynn recently launched the Soil Health app, which was funded by the Australian government's Natural Landscape Program, Smart Farm Small Grants, to provide essential information about the complex aspects of the soil health in a digital format. Wonderful. So I was recently 2 weeks in India on work, and it's just amazing. A lot of people looked at it. I sent the link, and they said, oh, can we use? And so you may get more inquiries from researchers from various parts of South Asia. So I encourage you to scan the QR code on the screen. Is there a QR code? Yes, to download that app. It's very simple, nice, and very easy to understand. I know, John Hicks, you're very much a fan of soil health, so we're doing as much, and Lynn is turning all that into a book, right? So it will be published soon. Many awards and achievements in 2021. Lynn was named as the inaugural recipient of the prestigious General Jeffrey Soil Health Award. Lynn will be chairing this session. Please welcome Lynn.
Hello, everyone. So thank you, Professor Siddique, for this introduction. Our first postgraduate presenter following afternoon tea, I hope you all had a nice little break. Sajeevee, got to pronounce her surname here. Sarah Chandra?
Yes.
A bit slow, I should say it more quickly. From the UWA School of Agriculture and Environment. Sajeevee is a final year PhD researcher. She obtained her Bachelor of Agricultural Science degree with specialisation in biotechnology from the Wahamba, I don't think I got that right, University of Sri Lanka. She then received her Master of Philosophy degree from the University of Perennial, Sri Lanka also, where she studied integrated water resources management, and she had a fellowship at the University of Wageningen during this time. Prior to coming to UWA, she worked as a scientific officer in the Sri Lankan National Research Council and as a lecturer in the Spectrum Institute of Science and Technology in Sri Lanka. Her main research interests are ecotoxicology, plant physiology, and biotechnology. Today, she'll be exploring the topic, remediation of iron ore mine tailings by growing perennial rye-grass with organic amendments. Welcome. Thank you.
Thank you, Professor. Good afternoon, everyone. I'm here to give you a talk on remediation of iron ore mine tailings by growing perennial ryegrass with organic amendments. First of all, I would like to thank my supervisors, Professor Zed Rengel and Dr. Zakaria Sulaiman, and as well as I would like to thank Australian Government RTP Scholarship Program for funding my PhD research, and also Institute of Agriculture for giving us this platform to present our research findings. So iron ore mining industry in Western Australia plays a significant role in resource sector. It accounts around 47% of the resources of total value of total resources in Western Australia in 2016. It generates around 640 million tons of tailings in Western Australia per annum. So the tailings are the material remaining after extraction and purification of ore. So why these tailings are problematic? Are those so important? Yes. The components in the tailings are spread to the surrounding areas and cause wind and water erosion. Most of the time, tailing has inadequate physical structure, nutrient content, and low cation exchange capacity, low water holding capacity, low microbial activity, and most of the tailings are comprised of higher concentration of heavy metals. So we need to rehabilitate in order to mitigate the negative impact on surrounding landscape. So the rehabilitation that we have, and most of the industries are adapted to, are physical methods. Under the physical methods, we have covering with new soils, electroremediation, and heat treatments. The problems of these physical treatments are they are very costly and labor-intensive programs. Some of the mining industries are adopted with chemical rehabilitation processes as well. They are comprising of leaching and acid extraction, and sometimes they use soil washing as well. Again, the drawbacks are costly and they improvise the physical characteristics. Novel methods at this moment, and most of the iron industries are adopting, is a biological method. It comprises of using of microbes, plants, and organic amendments. Sometimes they are using separately, or maybe combining together they are using it. Thing is that they are very cost-effective, viable, and eco-friendly. So we understood that the strategic way to remediate the iron ore mine industry and tailings are application of organic amendments of the plants. For that, we use compost and perennial rye-grass. So because they are very low in cost and low in maintenance, long-term, viable, and eco-friendly. The first aim of our study is to investigate whether we can improve the physicochemical characteristics of tailing by using compost. So we use 2 iron ore mine tailings from Pilbara region, and we combine tailings and compost according to these treatments. The first treatment was 100% tailings, whereas we didn't use any compost. Second one is 50% compost application. Third one is 75% compost application. The fourth one is 100% compost, which means has a positive control. So these are the physicochemical characteristics that we investigated on. First, we consider about the particle size distribution, sand, silt, and clay, pH, which is the most important physicochemical characteristic of any soil or even tailings, and the cation exchange capacity, total organic carbon, and water holding capacity. We understood that the initial, the original level of both A and B tailings has low cation exchange capacity, low total organic carbon, and low water holding capacity. Fortunately, the tailings I got to investigate had near neutral pH. If you see the silt and clay percentage in tailing A, it has higher percentage of clay and silt in tailings A, whereas in tailing B had high percentage of sand. With the application of compost, we understood that all the characteristics we investigated here improved. Then we wanted to find out whether this application of compost is impacting on total metal concentration of tailings. The metals that we have investigated here are arsenic, iron, lead, and zinc. You all can understand initially higher concentration of arsenic could be seen in tailings A. Higher concentration of iron could be seen in both tailing A and B. High concentration of zinc could be seen in both tailing A and B. And also the lead is also very high in both tailing A and B, but it is a bit lower than the permissible level. But with the application of compost, total metal concentration was reducing, whereas the other physicochemical characteristics was improving. The second aim of this study was to investigate whether this application of compost is improving the biological characteristics. Among many of the biological characteristics, we consider the microbial respiration. Microbial respiration is one of the most sensitive parameters which gives you an idea about how healthy your soil is or your growth substrate is. This is an incubation study. We did over 105 days period. We understood that initially there's a slight change with compost application, but in 85 days, after 85 days of incubation, the microbial respiration increase in unamended tailings, and higher could be seen in 75% compost application. With that, we understood that biological characteristics such as microbial respiration could be also improved with application of compost. And also, we thought that this is quite favorable for the plant growth and microbial activities. The third aim of our study was to investigate whether we can promote and maintain sustainable plant growth in mine tailings. For that, we use perennial rye-grass, the Lolium perenne. It's under the Poaceae family. We use Australian inbred line, which is improved for long summers and low fertility. We grew the plants over the period of 45 days and 60 days, and this is what we got after the time period under the glasshouse condition. You could see that higher growth could be seen in 60 days growth cycle than the 45 day cycle, and of course in 75% given you the higher plant growth, and tailing B had a proper growth than tailing A. Now we will see what are the how the growth parameters are contributing to this outcome. From this point onwards, I will be more focusing on 75% compost application in both tailing A and B at 45 days harvest and 60 days harvest. First, we will look at how the shoot dry biomass is changing. Both tailing A and B had 98% increase with application of 75% compost at 45 days harvest, whereas in 60 days, it is 99% increase and 50% increase. So we understood that 75% compost application is giving you the best shoot dry biomass in both tailings in both harvest times. So we wanted to see how these below ground parameters are, growth parameters are contributing to that particular results. First, we will see how the root dry biomass is changing. According to the results, both tailings A was given 84% increase, and tailings B was given 25% increase in 45 days. Whereas in 60 days, it was 95% and 39% increase with application of 75% compost. And also we consider about the root architecture. We, with the Windriser software, we scanned the roots with Windriser software. This is what I got for total root length. We understood that at 45 days harvest with the application of 75% compost, it's 65% increase. In 60 days harvest, it is 44% increase. The most important other root architecture parameter is the root diameter. We understood that in tailings A, it is 90% increase with 75% compost application and 57% increase in tailings B in 45 days harvest. In 60 days, it was 95% increase and 60 days increase again in tailings A and B respectively with application of 75% compost. Not only the root architecture, we consider about the mycorrhizal colonization. You can see that initially mine tailings, unamended mine tailings doesn't have much mycorrhizal colonization, but with application of 75% compost, it is 99 increase could be seen in both harvests. So 75% compost application was given us the best root dry biomass, root length, root diameter, and as well as mycorrhizal colonization in both tailings in both harvests. After having this good and proper growth in the perennial ryegrass, we wanted to see whether that will impact on the substrate metal concentration or whether this perennial ryegrass could remediate the metal concentration in substrate. For that, first we consider about the accumulation in zinc. accumulation zinc in the shoots. We understood, you can see that in 45 days harvest is a consistent metal accumulation could be seen in 45 days, but in 60 days, in 53% increase and 7% only 7% increase was seen in tailings A and B respectively. In copper, 62% increase was observed and 43% observed in tailings A and B respectively, whereas in 60 days it was 63% and 35% increase with the application of 75% compost. But lead is totally different behavior we could observe. You could see in 45 days harvest, it was decreased with 75% compost application. In tailings A, it was 76% decrease, whereas in tailings B, it is 41% decrease. But unfortunately, we could not see any metal accumulation, especially the lead accumulation in 60 days harvest, it is below the detection limit. Same trend was observed in iron as well. In 45 days of harvest, it was decreased by 80 to 85% and 69% decrease in tailings B after application of 75% compost. In 60 days harvest, it was 93% decrease. In tailings B, it was 76% decrease. So we could summarize the all results is that 75% compost application improved tailings physiochemically and biologically. The promising perennial rye-grass could be grown and seen in tailings with amended compost. Added compost immobilized iron and lead because it's making as a lattice structure underneath, whereas in copper and zinc, it increased with compost application, which means compost is giving and contributing more soluble carbon to the medium, so that soluble carbon is making complexes like metal organocomplexes, and it enhances the growth through the roots to the shoots and accumulate in the shoots. So from this study, we understood that under the glasshouse conditions, there's a promising results that we can have the remediation or rehabilitation with applying compost to the mine tailings. But we need to disseminate this result to the field condition, see how it is actually working on the field conditions. Thank you.
Thank you for your presentation. Do we have any questions? Yes.
Have you considered using biochar as a supplement along with the compost?
Well, this experiment actually only the compost. My other experiment is based on biochar, which I didn't include in the presentation. That is also, of course, another different results that we obtain compared to the compost. But we didn't mix together compost and biochar to see whether there is any synergetic effect on metal accumulation and growth of perennial ryegrass. Yeah.
Thank you. That was very. Oh.
Who's got the speak? Don. Don.
I've got the speak. Yeah. Very interesting talk. Thank you very much. I'm thinking about industry application. A lot of our iron ore mines are very remote from a reasonable source of compost, and presumably it would cost a lot to get it there. And secondly, I mean, you've got this great mountain of tailings. Do you have 75% in the top 10 centimetres of tailings or the top 20 centimetres of tailings? How would you do that practically?
Well, actually, I didn't put the, this is actually a glasshouse experiment, first thing. Second thing, I didn't lay on top of the tailings, I mixed it with, because, you know, in the, of course, when it comes to the field conditions, it's really hard to maintain the certain depth or height of compost along the tailings. What happened in the industry is that they are just dumping into the open area, and when you are going to rehabilitate, you have to level the land. So when it comes to leveling, I don't think that you could maintain a proper depth or leveled land. So that's why I mix the tailings with compost. The first part of your question that you said that the cost will be a problem. Yes, it's kind of a concern in this rehabilitation process, because any of the rehabilitation process going on, you'll be incurred with the substantial cost of substantial initial cost of rehabilitation. But when it comes to the industrial level and the money that they are generating, the revenue that they are generating per year is huge when it compares to they are allocating some money to the rehabilitation process. Actually, most of the mining industries are at the moment investing more on rehabilitation researchers, and as well as they're adopting it as well. Thing is that it takes time, but you need to do. Yeah.
John? Oh, sorry, we've got one up the back. Okay.
Thank you for that presentation. Why did you use, or what do you propose to use, rye-grass? And do you think that that could be a risk to the native flora ecologically, like introducing systematically that species?
Yes, actually, it's a very good question, because when you come to this iron ore mine or any other mine tailings, first thing you have to think about the rehabilitation, and then you have to think about whether we can have a productivity. That's a later stage. When it comes to the selection of the plants, fortunately, I have the near neutral pH in the tailings, so I don't want to worry about the pH ranges that when selecting the plants. But most of the time, some of the mine tailings are having, some of the iron ore mine tailings are having low pHs, which is acidic. So when it comes to the pH ranges in the iron ore mine tailings, having higher pH range from acidic to alkaline. So in that case, perennial ryegrass is having good adaptability for the pH range, one thing. Second, I consider the Pilbara region where the mining arena of Western Australia having extreme environmental conditions. So the perennial ryegrass I use here is an improved variety, so which means it can sustain in the low fertility and the long summers. Because in the summers, in Pilbara region, normally the temperature goes up to more than 45 for many days, and in the winters also same, you can have flash floods and severe thunderstorms. So those type of environmental conditions, considering those type of environmental conditions, we believe that this improved variety will sustain. Yeah.
But that very strength won't be a problem for the local flora?
Well, again, when we consider about selection of the plants, we have a what you call grasses biosurvey. At the moment in Western Australia, they have quarantine restrictions for the grasses. We selected 2 grasses, but unfortunately one is left out because of the quarantine purpose. At the moment, perennial ryegrass is actually considered as invasive, of course, but not the perennial ryegrass, it's annual ryegrass.
Thank you. John?
No, my question was why did you choose ryegrass? I would have thought you'd do better to look for a species that was local and lead tolerant.
That's one thing.
So could I just add to that? So is the intention in the long term to introduce other species, plant species but to get that started with the ryegrass?
Well, it has to be because actually initially we had a concern of mix of 2 grasses, like perennial ryegrass and Miscanthus sinensis, which is Chinese silver grass. It's actually intermittently we could find in Western Australia, but is not permitted. So if you are going long term, maybe we can have a mixed cultures, like mixed cropping system with perennial ryegrass with some other buffer grass or something. But initially, we think that the perennial ryegrass would be the better to have initiate the rehabilitation.
Thank you. Well, thank you for your presentation.
Thank you, Professor.
We think you've tackled a very difficult problem of this rehabilitation in this extreme environment. So wish you all the very best. And if you'd like to join me in thanking you. Only one. Thank you very much.
Thank you, Professor. Thank you very much.
So our next presenter is Barya. I'll get her name wrong as well. I'm sorry. I apologise. Siddique just said that. Can you say it again?
Disanayaka.
I don't need to say it now. I apologise. From the ARC Centre of Excellence in Plant Energy Biology, which is located in this building.
In this
building,
is that right?
Yeah. Yes. In 2017, she completed her first-class honours degree in plant biotechnology in Sri Lanka. She has had a fascination of how plants withstand harsh environmental conditions, and she's had a keen interest in proteometrics, which you might like to explain to us. And this led her to pursue higher degree studies in biochemistry. In 2018, she received a GRDC scholarship to start a PhD with Professor Harvey Miller in his lab upstairs. And during a PhD, she studied physiological and molecular level responses of wheat roots under salinity stress conditions. She successfully completed her PhD in March this year, congratulations, with publications in leading European proteometric journals. With her expertise in plant proteometrics, she is currently working as a research associate in the same laboratory in an Indian-Australian collaborative project on enhancing drought tolerance in chickpea, which I think yesterday you were at the seminar yesterday?
No, I was
not. Oops, sorry. But it was a very exciting day yesterday, I understand. So the title of your presentation is up here. And I'd like you to join me in commencing. Thank you very much.
Thank you, Professor Anna. Hello, everyone. I'm Bhagya Disanayaka. So today I'm going to reveal some of the hidden adaptations of breadwheat roots under salinity stress. First of all, I would like to acknowledge my supervisors for their guidance, Middle Lab group members for their support, and the GRDC for funding my project. Salinity is one of the major abiotic stress constraints that affects the crops throughout the world. More than 1 million hectares in agricultural land in Western Australia is severely affected by salt. So if you look at the picture on the left, you can see patchy growth of crops due to the saline conditions. And on the right-hand side is a picture where the land is severely affected by salt. So the loss of agricultural productivity due to the salinity damage is estimated to be worth at least $519 million per year. When we consider wheat, it's a moderately salt-tolerant crop, and we need to improve the salt tolerance of wheat in order to sustain the food production for this growing population. There have been lots of traditional breeding approaches trying to produce more and more new salt-tolerant wheat varieties, but they have been only focusing on above-ground changes and achieved limited success over these years of time. So why do we think roots is important? Because roots are the first organ that actually perceive the salt. But so far, roots have always been an underutilized source of crop improvement. So we think we need to better understand the root responses in order to utilize the roots for the improvement of salt tolerance in wheat. So today I'm going to show you how we use proteomics to discover the salt stress-related changes in the metabolism of wheat roots. Studying the proteins is important because proteins are the key players of metabolism, and they are the ones that determine the phenotype of the plant. When we look at the wheat root system architecture, wheat root system consists of 5 to 6 seminal roots, and then the lateral roots arises from these seminal roots, and in the later stages of development, it produces the nodal roots. So what happens under salinity is that there's an overall reduction in the root length of the axial roots. This was known for a long time, but then the metabolic reasons behind this growth reduction was largely unknown. So my PhD project was designed to see how the wheat roots respond to salt stress. So in the first half of my PhD, I conducted a study where I compared mature root and root tip responses to salinity in wheat variety Scepter. Scepter was chosen because it's a leading wheat variety grown in Western Australia, and to grow the plants, I used an unsupported hydroponic system. So this hydroponic system allowed me to have so many plants within a single tank. Also, it was really easy to harvest the roots without damaging them. So at the emergence of the third leaf, I started adding the salts. So I gradually increased the salt concentration up to 150 millimolar, and then I harvested mature roots and root tips after 3 days and 6 days of exposure to the final salt concentration. Then we measured a range of physiological parameters, including root dry weight, shoot dry weight, and oxygen consumption rates. So what we saw was that after 6 days of salt stress, there were significant reductions in number of physiological parameters such as root dry weight, shoot dry weight, oxygen consumption rate of the root tips, as well as the total root length. So what does this mean? This means that there's a negative impact of salt stress on the root growth and respiration. But we all know Scepter has some kind of salt tolerance in the field because it's enabling reasonable harvests. But then our research suggested that the early vegetative state of Scepter is significantly inhibited by salinity. So then we wanted to see what kind of metabolic reasons are underpinning this physiological sensitivity. For that, we conducted a proteomic analysis in mature roots and root tips. So with the proteomic analysis we did, we were able to quantify 647 proteins in mature roots and 737 proteins in root tips. So among these proteins, then we wanted to see what kind of proteins were salt responsive. For that, we conducted a differential abundant protein analysis. And this particular graph shows the number of salt responsive proteins in mature roots and root tips after 3 days and 6 days of salt stress. So if you look at the 6 days time point in root tips, you can see that most of the proteins were decreased in abundance compared to the number of proteins that were increased in abundance. So what does this mean? Does it mean that the root tip metabolism is more affected under salinity? Let's find out. So then what we did was we put these proteins into pathway context. Then we saw there are some distinct changes in the metabolism of the mature roots and root tips. So in the root tips, most of the proteins that are involved in carbohydrate metabolism, which simply means that are the proteins that are involved in interconversion of starch to sugar, they were increased in abundance, but in mature roots, those were decreased in abundance. But in root tips, most of the proteins that were involved in energy metabolism, amino acid synthesis, and protein synthesis, they were decreased in abundance. So overall, this means that there's a negative impact on root tip metabolism. And this study actually provides metabolic evidences for the root tip growth reduction. But actually, it did not provide any clue whether roots have better adaptations to salt. So to answer this question, we conducted another study with 4 different wheat varieties, which differ in their level of salt tolerance in shoots. So before moving on to the changes that we observed in those 4 wheat varieties, I would like to give you an overall idea about the salt tolerance mechanisms in wheat. Wheat has 2 different salt tolerance mechanisms. One is sodium ion exclusion. So by sodium ion exclusion, it prevents sodium ion accumulation up to toxic concentration within the leaves. Almost all red wheat varieties have some kind of sodium ion exclusion capacity, but then these Westonian X1 and X2 lines, they have enhanced sodium ion exclusion capability due to the presence of NEX1 and X2 genes. There's another important salt tolerance mechanism called tissue tolerance, and this is achieved by compartmentalization of sodium ions within the vacuoles of the cells in the leaves. So usually the tissue tolerant wheat varieties, they have more sodium ions within the shoots of the plant. So Mochodi espica branca is a good example for the tissue tolerant wheat variety. And there was also another reason for selecting Gladius and Mocho, because recombinant inbred line populations has been developed in Adelaide using this Gladius and Mocho as parental genotypes. We measured a range of physiological parameters in shoots and roots under salt stress conditions in these 4 different wheat varieties. In terms of the shoot ion content, so you can see here the sodium ion content in the shoots of these 4 different wheat lines under control and salt treatment conditions. So what we see is that there's at least 3-fold more sodium ion accumulation in mocho when compared to Gladius, Westonian X1 and X2. And these Westonian X1 and X2 lines, they maintain significantly lower sodium ion content compared to the other two varieties. In terms of the potassium ion content, we can see that Gladius maintains the lowest potassium ion content compared to the other three varieties. And also, so this is the potassium sodium ion ratio, which is an important measurement in the interpretation of salt tolerance level of a particular variety. So we can see that Westonian X1 and X2 lines, they maintain significantly higher potassium sodium ion ratio, which means that they can discriminate the potassium over sodium ions, which is beneficial for their growth and development under salt stress conditions. In terms of the root ion content, we did not see any significant changes among the genotypes under salt stress conditions in terms of sodium ion content as well as the potassium sodium ion ratio. We also measured some of the shoot physiological characteristics under salt stress, such as chlorophyll content and rate of photosynthesis. So what we observed was that under salt treatment conditions, Westonian X1 and X2 lines, they have higher chlorophyll content compared to Gladius and Mocho. Also, they maintain significantly higher rate of photosynthesis compared to the other two varieties. So what does this mean? It means that these Westonian X1 and X2 lines, they have higher photosynthetic capacity under salt stress, which is beneficial for their growth and development. And also we measured some root growth characteristics such as root dry weight and total root length. What we saw was that Gladius had the lowest root dry weight and lowest root length compared to all the other wheat varieties. So this means that the root growth of Gladius was most affected under salinity. So overall, our physiology data suggested that Gladius is the least tolerant cultivar, while Mocho, Westonian X1 and X2 lines, they are more tolerant than Gladius. So then we did a proteomic profiling in roots of these 4 different wheat varieties. So with that, we were able to quantify 3,784 proteins in roots of these 4 different varieties. So among these 3,000 proteins, we wanted to see what kind of proteins were salt responsive. For that, we conducted differentially abundant protein analysis. So this particular Venn diagram shows the overlap between the salt responsive proteins in Gladius, Mocho, and Westonian X1 and X2. There you can see 139 proteins were common to all 4 wheat varieties. And if you look at the edges of this Venn diagram, those are the proteins that are unique for each of these varieties. So now you must be thinking, do these varieties have similar responses or do they have differences in their responses? So to answer this question, apart from the differentially abundant protein analysis that we did, we conducted the 2-way ANOVA analysis in order to identify the proteins that are significant for the genotype, treatment, and the genotype treatment interaction. So overall, our proteomic data suggested that there's a common metabolic bottleneck happening following the salt exposure. So before diving deep into this metabolism, I would give you an overall idea about plant metabolism. So plant metabolism can either be primary metabolism or secondary metabolism. So primary metabolites, they are directly involved in the growth and maintenance of the plant. And in terms of secondary metabolites, they are involved in stress-related defense. So our proteomic research suggested that the roots have changes in both primary and secondary metabolism. Imagine if this is a root cell, so what happens inside the root cell is that glucose or the 6-carbon sugars get converted into pyruvate or 3-carbon organic acids. So this process is called as glycolysis. And then this pyruvate goes inside to the mitochondria, which is the powerhouse of the cell, and then get converted into other organic acids. And this process is we call as TCA cycle. And there's another very important process happening in the membrane of the mitochondria, which is known as oxidative phosphorylation. So glycolysis, TCA cycle, and oxidative phosphorylation comes under primary metabolism. What happens under salt stress is that salt stress increases the energy demand inside the root cells. So these glycolysis, TCA cycle, and oxidative phosphorylation, these pathways can generate energy in the form of ATP. And then this ATP can be used to fulfill the energy demand within the root cells. It has been also known that under salt stress conditions, the cell wall integrity is negatively impacted. So the plants have a mechanism or pathway called shikimate and phenylpropanide pathway, which allows the plant to produce lignin in the cell walls. So the lignification of cell walls can help to maintain the cell wall integrity. This pathway is involved in the secondary metabolism. So now you must be thinking, how my 4 different wheat varieties come into this pathway, how do they fit into this pathway context? So the salt-tolerant Westonia next 1 and next 2 lines, they significantly increase the abundance of proteins involved in glycolysis, TCA cycle, and oxidative phosphorylation, which means that they are trying to generate more and more energy under salt stress conditions, which is beneficial for their growth and development. And it has been also known from previous research that salt stress inhibits the pyruvate metabolism. So our sensitive cultivar gladius, it panics out and then it tries to convert pyruvate into other metabolite forms and try to reconfigure its metabolism downstream to pyruvate. Our protein data also suggested that regardless of salt stress, the salt-tolerant wheat lines, they have the capability to produce more and more lignin. So when the salt stress appears, they can still maintain their cell wall integrity and grow and continue to grow and develop. So overall, what this suggests is that the roots of the salt-tolerant wheat lines, they have better metabolic adaptations compared to sensitive wheat line. In summary, mature roots and root tips, they show distinct responses under certain stress conditions, and the roots of the tolerant wheat lines, they have better metabolic adaptations by either constitutively producing more lignin or by inducing energy production. Also, in our study, we identified putative biomarkers that can be used to screen breeding populations in order to screen and identify the wheat lines which have better adaptations of roots to salt stress. There comes to the end of my presentation. Thank you all for paying attention.
Thank you very much. Do we have any questions?
Thank you, Bhagya. A wonderful presentation and good study. Just a comment, Westonia is bred and developed in Western Australia. So naturally the breeders must have been testing under natural salinity conditions, not severe, but most of our land has got some sort of salinity. Whereas Gladius is coming from Eastern Australia, you may correct me that. There the problem is not there. So you have any suggestion about that? So unknowingly the breeders have been doing that. If they don't do well, they won't select for yield.
So you mean, why did I selected these
Yeah,
lines?
because these were bred and selected by one of our former colleagues. He's retired.
Yeah, so
Robin, not Robin, will come. Robin Wilson has developed a Westonia.
Yeah. So these 4 different wheat lines, they have different salt tolerance mechanisms, and but so far no one has actually looked at the root, how the root actually contribute to these salt tolerant phenotypes. So that's why we wanted to select wheat varieties that have different salt tolerance mechanisms, and we try to find out how the roots contribute to this particular phenotype. And also Gladius and Mocho were selected because in Adelaide they have already developed wheat recombinant inbred lines using these 2 genotypes as parents. So initially we wanted to test our putative biomarkers with this recombinant inbred line populations. Yeah, that's why.
Could I just follow that up with a little question? I know you've grown these in hydroponics, but do you know anything about the exudates that go into the soil or into the solution from these different cultivars? Is there a difference in the exudate, the carbon going into the soil, which would feed microbes?
Actually, I haven't analyzed any root exudates in these studies. Yeah.
But it could affect the microbiology of the soil.
Of course,
and have a feedback. Anyway,
yeah, but I haven't looked at that in my study. Yeah.
Thank you. Where's the microphone? Yes. Any other question? Yeah. Thank you.
Yep. So just a question, probably more of a technical question, but in terms of the level of salt that was used in the first study with the scepter, how does that relate to the concentrations that we'd normally find out in the field?
Yeah, so the final salt concentration that we were using was 150 millimolar, and that was, I mean, in all the literature, previous literature, we have seen that people have been using this particular concentration, which is similar to saline-affected fields in most of the lands in Western Australia. Yeah.
I mean, second part of the question was, scepter was used in the first part of the study.
Yeah.
Was there a reason why it wasn't used with the 4 varieties at the the second part?
We used scepter to see how the root responses differ in the mature roots and root tips. And then in the second part of the study, we wanted to analyze 4 different wheat varieties which have different level of salt tolerance. Actually, Gladius, we think that Scepter and Gladius should have some kind of similar level of salt tolerance because they don't have specific mechanisms rather than just a sodium ion exclusion. So, yeah.
Another question? Maybe I'll ask another question. In Western Australia, we went through a period of declining rainfall, and the salt problem became even worse. And now that's changed a little bit. How is the impact of the choice of a wheat plant grown in Western Australia? Has it been changed in response to changes in the rainfall?
Changes in the rainfall. Actually, so these Scepter, they have been developed from previous wheat varieties which have been showing good yields under saline affected lands. So they are keep breeding for maintaining that. Yeah.
Okay. I don't think we'll recover totally from problems of
problems.
salinity, but you're working with the India project in joint Indian Australian.
But then it's on chickpea drought tolerance
in a different plant.
In different plant.
But so is there how do you link your expertise in what you've done with wheat into that space?
So this untargeted proteomic analysis, what we have did here, it is kind of similar what we do in the chickpea project as well. We are trying to find out purity biomarkers that are associated with the drought tolerance, and also in chickpea project, it's a root associated marker. So there's a focus on roots and proteomics, untargeted proteomics. So I can apply my knowledge which I have gained through my PhD directly to that.
So that's wonderful to hear. Thank you. Thank you. Are there any other comments? Yes, sorry.
A couple of comments. Looking at that photo on the screen there, did you look at those couple of green patches to see what the difference in the soil was there?
Sorry.
Is that where there's accumulated drift or where a sheep died or?
No, those are, so this is the salinity affected land and there's a patchy growth because of this severe salinity condition. So there's high salt concentration in some of the parts of the land. So there's a patchy distribution of the growth.
And the second question, if I may, it is well known that active soil biology can exclude certain elements from the plant. Have you looked at any biological treatments to see whether this is effective? And in all these cases, we're talking about soils that are just to that marginal salinity where things might grow. If you've got the visible salt encrusted on the surface of the soil.
You mean elemental analysis? You mean elemental analysis or something like?
But because a well-known regenerative farmer is finding that there's patches of soil like that are re-establishing and growing crops after the application of 2 or 3 years of biological extracts.
So you, is your question about whether I have analyzed any elemental analysis in my studies, is it?
I think that the question is beyond what you've actually done in your study.
Yeah.
So because the studies that you've been analysing under pure conditions didn't relate to soil amendments. So it's another part of
it's another part of which we haven't looked at in my study. More work to be done.
I'll let you off that one. Okay. Thank you so much for your presentation.
Thank you.
Anne. Thank you very much. Well done. So we have one more. So you might have realised that one of the speakers earlier was not able to come previously, just prior to afternoon tea. So our next speaker is Isabelle Sewell, who's from the School of Biological Sciences at UWA and the UWA Oceans Institute. So Isabelle grew up in Perth, where her love for the ocean from a young age, led her to pursue a career as a marine biologist. Through her master's and now carried through to her PhD, she's developed a keen interest in the application of circular economies. with a particular focus on the relationship between future protein and aquaculture nutrition. Isabelle's research is funded by the Fisheries Research and Development Corporation, and she has received a DPIRD Fish Aquaculture Scholarship to do her research. And in 2020 and 2021, she had an award, the Alan Robson and Robertson Award. That's excellent. So her presentation is entitled, there it is, someone's put it up. So thanks, Isabelle.
Thank you. So I just wanted to start by acknowledging that this research wouldn't be possible without the funding that I've received through the FRDC grant, with contributions from the Department of Primary Industries and Regional Development, Chem Centre, Future Green Solutions, GEA Engineering, and Ridley Agri Products. So for some background, fishery resources are currently in decline for a number of reasons. And if we pair this with the global population expecting to exceed 9 billion by the year 2050, it places this huge pressure on food sectors to maximise their production whilst conserving the world's remaining resources. In theory, aquaculture is a good solution to that because we're farming fish instead of removing them out of the ocean. However, in practice, that's not necessarily the case, as many aquaculture species, especially within Australia, are carnivorous. So they have high dietary protein requirements that are often delivered in the form of wild-caught fish, which we refer to as fish meal. So we find ourselves in this negative feedback loop, and in order to break away from it, we need to find alternative protein sources that still enable growth whilst maintaining species well-being. So my PhD has focused on assessing whether black soldier fly, and specifically their larvae here, are a good alternative protein source in aquaculture diets. Now, there's many benefits associated with the commercial production of these larvae, and one of which is the promotion of a circular economy, which is depicted in this infographic. So first, we have an input of an organic waste. So this could be things like fruit and veg that are considered too unaesthetically pleasing for supermarket sale, as well as things like manure. And then the larvae feed on these waste products and bioconvert that into their own biomass. So this means that their diets are controllable, but also to an extent we can manipulate the nutritional profile of the product at the end. From this, we can derive a number of products like protein, oil, as well as utilize the frass. And then we hope to feed that back into other agri and agriculture sectors to complete that circular economy. So I'm most interested in the protein product because it's obviously quite high in protein, but it's also high in fats at levels that are comparable to fish meal. So the component of my research that I'm presenting on today aimed to assess the effects of black soldier fly larvae on juvenile barramundi diets. So I did that by feeding 8 experimental diets to barramundi over a 13-week period. And using those 8 diets, I wanted to ask 3 questions. So the first one was, how does an increased inclusion level of defatted black soldier fly affect the fish? And we did this with the hopes of determining an optimal inclusion level. So our first diet here is the control, and that's modelled off a commercial barramundi diet. And the following 4 diets contain graded inclusion levels of defatted black soldier fly in substitute to what I refer to as other proteins. So these are things like soy and wheat-based products. The second question was, did it matter if we used a defatted protein or a full-fat protein? So there's a lot of variability between the results that have been published in aquaculture-based studies using black soldier fly, and this could be attributed to a number of reasons, but one of which is that research groups either use a defatted product or a full-fat product. No one's actually looked at how the 2 different products potentially perform differently. From a commercial point of view, it's ideal to use a defatted product because the protein can be utilised in one industry, so in this case aquaculture, and then the oil could be utilised in a second, so for example, biofuels. The last question I wanted to ask is, does it actually matter what we substitute the black soldier fly with as well? So all studies so far have focused on substituting with fish meal, but we know that fish meal is incredibly important in aquaculture diets. So I hypothesized that the potential removal of fish meal was actually masking the benefits and the performance of black soldier fly. So at the end of the 13 weeks, I found that there was no significant difference in growth between the 3 diet combinations. However, I just did want to point out that there was significant growth over time. So the fish went into the trial at about 35 grams, which is about this big, and then ended on an average of 400 grams, which is about this big in just 13 weeks, which was great. At the end of the trial, we also considered the feed conversion ratio, also known as an FCR. So this is incredibly important to consider both from a biological point of view as well as an economical. So it tells us the biomass in a ratio to the biomass out. So in this case, how many kilos of feed are required to produce 1 kilo of edible fish. The results I obtained are up in this bar graph, and you can see there's a little bit of variability with the results. However, these are the values you would expect to see for an FCR in other agricultural sectors, including the fish sector, for salmon specifically, and these are the values obtained in ours. So the lower the FCR value, the better. So this is a great start for the inclusion of black soldier fly. So for these results so far, it's suggesting that black soldier fly is a suitable protein alternative. However, there is a caveat that this growth trial only ran for 3 months. So growth trials are really good indicators, but they're somewhat limiting. So in my case, I could only run the trial for 3 months before the fish became too big for the tanks, and that became a confounding factor. But there are some biological factors that we can take into consideration at the end of the trial, and they can infer over time whether the inclusion of black soldier fly would be beneficial or not. So we already have an idea of growth, but on top of that, if we consider biochemical health in the form of blood, gut health in the form of histology, immune response through a bacterial challenge, as well as consumer perception through the nutritional profile of the final fillets, we get a better idea of the degree of suitability of the black soldier fly as an alternative protein source. So starting with blood health, we analysed a number of blood parameters. Only 4 returned a significant result in terms of the increased defatted inclusion. So AST, ALT, and GLDH, which is shown up here, are liver enzyme markers, and they're used to indicate cellular damage. So the higher the value, the more likelihood of damage. We found that as the inclusion increased, so did these values, but upon further analysis, it was concluded that this was actually just being driven by the 40% inclusion ratio. Cholesterol also returned a significant result. Cholesterol is an important component of the cell membrane, and it's a precursor of all steroid hormones. So the lower the value, the lower the health status of the fish. We found that as inclusion increased, the cholesterol rates decreased. Significant differences in cholesterol were also seen with defatted against full fat, with the defatted diets being significantly lower in cholesterol. And again, there was a significance with cholesterol with the presence against no presence of fish meal, with the fish meal diet being significantly lower in cholesterol. With the fish meal compared against no fish meal, there was also a significant difference in bilirubin, which is seen on the right here. So bilirubin is a compound that's found in the liver, and it plays a role in the clearance of cellular waste. So the higher the value, the higher the likelihood of impaired liver function, and we found that there was a significantly higher levels of bilirubin in the diets containing no fish meal. So to summarise that, even though there were significant differences, these are still within a healthy range. So instead of being immediately concerned at this point, we just need to be mindful of this moving forward. Then in terms of gut and liver histology, this is a histology slide shown from one of the guts in a fish at the end of the trial. And these are the villi tips here, and they're touching each other nice and close with not much gaps at the top, which is a good indicator of good health in the fish. And this was consistent across the 3 diet combinations. However, I just did want to point out that down the bottom here, this is what a normal villi tip looks like with the rich, deep purple staining. And then this is what a fatty villi tip looks like with the fat being washed out and staining as white. And this was a slide that was taken from a fish fed one of the full fat diets. So to conclude again, although this is still within the healthy parameters, we are a little bit hesitant of a full fat diet because there's potential that over time those fatty deposits could increase and cause impaired health to the fish, but at the moment it's not at a level we need to worry about. I also performed a bacterial challenge on the fish at the end of the trial. So bacterial challenges can be used as a vessel to assess the role that black soldier fly larvae may perform in the immunity and the immune response of these fish to diseases. So I injected the fish with a lethal dose of Streptococcus inii, which is a disease that affects warm water fish and is especially prevalent throughout barramundi aquaculture in Australia. So after injecting them with a lethal dose, we plotted their survival over time, which was just a 10-day period. And the only difference we found was that as defatted inclusion increased, there was a significant decrease in survival. But similar to the blood results, this was being driven by that 40% inclusion rate. So this is the first result where we actually are quite worried about the result, with that 40% inclusion level causing a reduced level of survival. Lastly, I also analysed the nutritional profile of the barramundi fillets. So this is incredibly important because nutritional information is relayed back to consumers in the forms of the nutritional facts that you see in the back of all packaged goods. And they allow consumers to make informed decisions on the products that they do or ultimately do not end up purchasing. So the values that I have up here on this nutritional fact sheet are the values obtained from our control diet. And there was no significant difference in protein and ash across the 3 diet combinations. And all the levels pictured here are the equivalent to a barramundi fillet that you're able to purchase from somewhere like Coles, which is great. However, we did have some significant differences in the 40% inclusion level of the defatted larvae in terms of the total fat. So unsaturated fats, monounsaturated fats, and omega-6s are what we consider to be healthy fats. And as inclusion increased, these fats decreased. This was also supported by the saturated fats, which are considered to be unhealthy fats, increasing with inclusion as well. So that's supporting each other there. So then we're a bit more hesitant again about that 40% inclusion level, with it causing an increase in bad fats and a decrease in good fats. So to bring you back to this infographic, I was interested in seeing how the application of insect protein can be used in the aquaculture industry in the hopes to fulfill the circular economy in this way. I've concluded that black soldier fly is a suitable protein source. However, the caveat is that a 40% defatted black soldier fly inclusion shows early signs of impaired health as well as nutrient availability. But we've also found that there is no difference between using a defatted or a full fat product, and it also doesn't matter whether we're substituting the black soldier fly with fish meal or general protein. Thank you.
Thank you. Do we have any questions? Microphone.
Maybe you want to go out of your comfort zone, and would you like to care to venture how you think perhaps the genetically modified omega-3 canola could have a role in fishery feeding within Australia in the future? A bit off your subject.
I'm not across my plant and plant-based agriculture, unfortunately, but I know that people have investigated plants as a protein alternative, but have been found to have anti-nutritional properties for what's been looked at so far and been causing inflammation of the guts in a few trials. So I guess it depends what the outcome of the genetic modification is on their ability to do that. But from what's been studied so far, my understanding is that it's causing quite a lot of inflammation and poor digestibility.
Very interesting talk, thanks, Isabelle. What are the issues involved in processing black soldier fly so it could possibly be used as an ideal fish food?
Well, currently the company I work for, which is Future Green Solutions, they've just become the first insect farm in Australia to be Feed Safe accredited. So the obstacles that you would face in using that is first, is it a safe product to use, which we've just solved with that one, but also there's the problem that feed companies would only adapt it if it is financially worth their time. So hopefully research like this is supporting companies to be more enticed to do that. And I know a few feed companies like Ridley Agri Products that have made my diets have begun to draw in insect protein for their own curiosity, and I think they're making small batches. But the few hurdles that they are becoming to be overcome in the recent months and years, so it's looking like quite a positive pathway.
I have two questions. The first is, what's the price of the black soldier fly? And the second is, has anybody tried thinking about turning them directly into a protein substitute?
For humans? Yeah. So I'm not too sure about the price. I know that Future Green Solutions are looking at doing economic modelling on it, and I'm not too sure where they're up to at the moment. But considering that they're taking a waste product that a lot of people pay to dispose of, and then can sell from just the one product, so the larvae, they can sell protein, fats, and the frass, which is the waste. So it's the dead carcasses, the uneaten substrate. There's a lot of avenues you can get out of that. So in theory, it makes sense that it makes a good economic model. In terms of eating it, it has to become feed safe accredited. And I know in Australia, being a Western country, there's a little bit of a stigma with consuming insects. But I will say when they're cooked, they do smell like popcorn chicken. So I think that, I don't think, yeah, if people are cooking them, it might be enticing. But hopefully one day, because I know cricket protein's taking off, but I think it's just maybe working through that stigma a little bit.
Isabelle, that leads on my question perfectly, actually. Did you taste the fish fillets?
Ironically, I'm actually vegetarian, so I haven't eaten any of this. But some of the guys who work at Future Green have, and they've got no comments. But I think, you know, if the strong is smell enough in a good way, it might be positive.
Thank you. Isabelle, I know there's often concerns about heavy metals from fish feed into fish farms.
Mm-hmm.
Have you looked at heavy metals in the...
I haven't, no. We are kind of we were going to look at it, but we're under the understanding that if we don't necessarily have a need to suspect that there's heavy metals in there, then we don't have a need to test it. But because the black soldier fly and the feed are made in commercial settings that are feed safe accredited, and we know that avenue, the streams are really well monitored in as a waste product for the larvae. It's not something that we have a reason to think that there is, because normally those heavy metals can be, like for instance, mercury can be passed from fish to fish. So yeah, at the moment, there's not a need for that, but it'd be something that would be cool to look at. And also the scope of whether black soldier fly can help potentially remove that from waste sources.
Thanks for the nice presentation.
Thank you.
One of the issues with this protein,
Mm-hmm.
the quality of the protein depends on the feedstock.
Yeah.
Using, so they are very concerned about contaminants getting into the.
Yeah.
Yeah, I know it's not part of the study.
Yeah. Well, Future Green Solutions once had an issue where because they take some poultry offal, and they were just recently fed a hormone, and then all the larvae died because they weren't aware of that. So that's allowed for them to increase through error, just being through a startup company just a couple of years ago, to be regulate that more and have more of an idea what's coming through. And then as part of their feed safe accreditation, that's really closely monitored now. So hopefully that's not an issue in terms of the companies that are accredited, but potentially on smaller farms.
Could I just ask quickly that you've made some predictions based on your work,
Mm-hmm.
if the fish got bigger?
Yes.
So is there a plan to put this at a commercial scale fish growing outfit?
I would love to do that for the, like in terms of running a trial that's larger. Yeah. So because they're a warm water fish, we've got a really great facility out at Shenton Park, if anyone's seen that, and there's some tanks that are 20,000 litres, but unfortunately it's not temperature controlled, so we can't put the barramundi out there. But I work with another girl doing her PhD called Kat, and she put some rainbow trout out there and could get them up to a larger size. So I think one of her trials ended at 1.5 kilos, which is definitely a table-sized fish. I'm not too sure of her results yet, but it looked quite positive, because at the end of the day with something like this, you don't have to grow a fish that's bigger. It just has to be the same size really for it to be competitive. And if it's bigger, that's a bonus.
Thank you. So I think we're done. So thank you very much.
Thank you.
well done. Thank you, Lynn, and again for chairing the session. And there's a little present for you as well. Thank you. I think we are on time. There's a few more things before we go out there and taste those barramundi. I think you all agree that the PhD candidates today did extremely well. They have worked very hard. I think many of them cancelled the normal student supervisor meeting saying that this is a high priority. And this was under the expert guidance of Professor Graham Martin. This is again his 16th year training and coaching. So please join thanking Graham. Then I just wanted to do a little bit more work again on Graham. So we're very pleased to share with you our upcoming event and encourage you to scan the QR codes. The printed version is also available, which is the Annual Industry Forum on Wednesday, 20th July at the University Club. No money involved. There will be drinks and food afterwards. This is again our 16th consecutive year. And an important topic, it's really timely, navigating the global agriculture marketplace in the Indian Ocean Rim, spotlight on India and Indonesia. So the keynote talk will be presented by former Minister for Foreign Affairs, and Defense as well, Stephen Smith. And we also got a wonderful speaker from India. Unfortunately, he won't come, but he will give a short but crispy video presentation recorded. We also got from the Indonesia embassy a person coming to talk about Indonesia. Then we have two farmers coming and talking their perspective, followed by Sundowner. And the opening address will be led by Honorable Mia Davis, again a wonderful person from country Western Australia. And in the same week, we have now celebrating the 50th year of Professor Graham Martin's 5 decades of association with UWA, including as an undergraduate, PhD student, and of course, and now he's the emeritus professor. A lot of friends, both within Western Australia, Australia, Europe, many decades of teaching, research, continuing teaching, and contribution to the sheep industry, and also the clean, green, ethical management of farm animals, established the Future Farm 2050 project, which is going quite well. So please look at that as well, and Phil is organizing. It will be a wonderful day at the University Club. So once again, I would like to congratulate the students. I think you all agree that very professional job. And this is again, a number of PhD students are sitting here. It's not easy, but it can be done with training, with careful. And this is really the stepping stone for you to get out there, because every day, if you are in the profession of research, have to give talk. Every day, someone will call you, and then you have to really say yes, can't say no to that. And this is really good. And we also thank Anna. Anna has to go, our Deputy Vice Chancellor of Research, for coming and giving those encouraging and complimentary words about postgraduate training, agriculture in this university, and more importantly in the state, and continued collaboration with our partners, industry, and other institutions. And I would like to invite you for the drinks and nibbles. I mean, that is the payback for you to sit such a long period of time. And thank you once again. But also I would like to thank my colleagues at the Institute. Rosanna, she's our communication officer. Cora is not in today. She's working from home. Then my two fellow associate directors, Phil Werko, you're here, Phil, somewhere? Yes. because of the drinks he come, and otherwise he won't come. And then Wallace crawled in, and again for the drinks. And Deb, our business manager, if you want to do anything to do with finance, go to Deb. And all of you, once again, thank you. And the chairs in particular, Hayley and Lynn. Once again, thank you. Let's go.